Next we have chromatography. Chromatography from the Greek word matlab ye hota hai, chroma means color and graphy means picture. Like through it's a technique where you separate inks, where you separate dyes, where you separate colors और किस basis पे according to the solubility. वैसे पीछे भी हमने जितनी separating techniques पढ़ी हैं हम उनको किन basis पे judge करते हैं कि भाई हमने इसकी distillation करनी है, इसकी filtration करनी है. हम यही देखते हैं कि भाई क्या ये चीज soluble है, क्या ये चीज insoluble है, क्या ये चीज soluble है. अगर इनसॉल्युबल है तो हम फिल्ट्रेशन यूज करेंगे, अच्छा सॉल्युबल है तो हम डिस्टिलेशन यूज करेंगे. यहाँ पे भी ये है कि ये डायज जो सेपरेट होंगे दे वुड बी अपॉन देयर सॉल्युबिलिटी. फॉर एग्जांपल इसका क्या मतलब है दैट द सेपरेशन ऑफ डाई कलर्स फ्रॉम ईच अदर अकॉर्डिंग टू द सॉल्युबिलिटी. मतलब कि जैसे हमने बात की फ्रैक्शन डिस्टिलेशन में. नाउ लिसन केयरफुली, व्हेन वी टॉक्ड अबाउट द कांसेप्ट ऑफ फ्रैक्शनल डिस्टिलेशन उसमें हमने क्या चीज पढ़ी थी. वी सॉ दैट इन फ्रैक्शन डिस्टिलेशन आपके पास जो डिफरेंट फ्रैक्शंस हैं they are separated on the basis of boiling points. वो जो separate हुई हैं they are being separated on the basis of boiling point. इसका lowest boiling point है up to the point where there is the highest boiling point. Same is the case here in simple chromatography. In simple chromatography as well, they would not be separated on the basis of boiling point. They would be separated on the basis of solubility. In fraction distillation, for example, different fractions were separated upon their boiling point. Lowest one first, highest one at the end. यहाँ पे क्या है in chromatography least soluble one the first and the most soluble afterwards. तो ये आपके पास chromatography में the dyes are separated according to the solubility. Their solubility will judge that at which point is their spot going to be formed. We also call this process as paper chromatography. So the apparatus we're going to use over here is first of all a piece of wood where this filter paper or the chromatogram जो पेपर होता है इसको या तो कुछ लोग फिल्टर पेपर कहते हैं और वी कैन आल्सो कॉल इट ए, यहाँ पे आई डोंट थिंक इट्स मेंशन, वी कॉल इट ए chromatogram. We call this a chromatogram, the paper on which this is being covered. Attached with a pin, ये आपके पास पेपर है, इस पेपर का नाम मैंने यहाँ पे बता दिया, it's known as a chromatogram. We have a beaker where the solvent is produced because you need a solvent as well. This beaker actually contains a solvent. It can be an acid as well, it can be water as well. यहाँ पे हमारा solvent जो है that is water, and this is the ink spot. Now I don't know which ink spot is this. For example, the ink spot is green. Ink spot कौन सा है? Green. Now green is the ink spot and I want to separate it. तो green किन दो colors में separate होगा? Into blue and into yellow. This is how we separate dyes, and they are being separated on the basis of their solubility. Now one of the things you have to note is that the baseline we also call it the pencil line. The baseline should be drawn with a lead pencil and not with ink pen, as the ink pen itself is a mixture of dyes and would thus produce an inaccurate result. जो आपके पास हम ये क्यों कहते हैं that we have to make the line with a pencil. आप खुद सोचें, if we make a line with an ink pen or a pointer or any other kind of a ball point, obviously that is also going to dissolve inside the solvent. Hence the line is made up of a pencil because pencil does not dissolve in water. Number 2, the solvent after drawing the baseline, this was your step number 1. Step number 2 क्या है? The solvent would rise up against the force of gravity to the pores of the chromatogram, and such movement is called capillary action. Capillary action कौन सी movement है? Capillary action is the action through which the dye actually moves up the chromatogram. आपने ink spot लगाया on the baseline. Now the capillary action will take place and the solvent would rise up along with the ink dye and spreading over and producing dots. Now this type of chromatography is called ascending chromatography. Then we have a descending chromatography. Over here the diagram is given of a descending one. ये आपके पास कौन सी है? This is your descending chromatography, whereas the chromatography shown over here is known as the ascending one. यहाँ पे the solvent is going to travel upwards. So ascending or descending में this is the process. Now in descending chromatography, the solvent moves downwards and it requires a longer chromatogram. The mixture spot should be neither too big nor too small for better separation and for good result. The solvent is the key factor for any type of chromatography. The solvent which produces maximum result separation from the mixture spot will be the right choice of the solvent. Now how to use the solvent? Whether should I use sulfuric acid, whether I should be using sodium chloride, aqueous sodium chloride, water use करूं, क्या करूं? Obviously the one which would be producing the best results the best dyes, the clear dyes, the most accurate results, the most precise spots, वो आपके पास best type of solvent होगा, and that would be the right choice of solvent that you will be using in your future chromatography experiments. Now for chromatography to occur, all the dyes present in the mixture spot should be moderately soluble. The dye which is the first to separate will be less soluble compared to that which will separate at the end. अब ये कैसे है, मैंने आपको शुरू में बताया that in fraction distillation, for example, everything would be separated on the basis of boiling point. But when we talk about chromatography, dyes would be separated on the basis of solubility. For example, if you read this point, the dye which is the first to separate will be less soluble compared to that which will separate at the end. जो पहले separate होगा वो less soluble है, जो कि बाद में separate होगा in the given solvent. And now how do we calculate? Now there's a thing known as the RF value which also determines the solubility of that specific dye. RF value हम कैसे calculate करते हैं? This is a very important formula and is also asked in MCQs and paper number 1 or 2 दोनों में ही आता है, theoretical B and multiple choice questions as well. So the formula for RF value is distance traveled by the dye and distance traveled by the solvent. अब ये दोनों कौन कौन से होते हैं? First of all, this is your pencil line. This is what? This is your pencil line, or what we call the baseline. Now the reading starting from the pencil line up to the solvent front. इस area को हम क्या कहते हैं? Where the solvent is ending. यहाँ तक, this is the finishing line known as the solvent front. Now the line extending, the length starting from the baseline till the solvent front is known as the distance traveled by the solvent front. This would be your denominator. ये क्या हो जाएगा? This would be your denominator. Now इसके बाद again starting from the baseline till the point where the dye, अब आपको specific dye दे देंगे. For example, बहुत से dyes होंगे, वो आपको बता देंगे that calculate the RF value for dye A, dye C, dye F. ठीक है? So you have to see that denominator or the distance traveled by the solvent front would be the same for all dyes. सारे dyes के लिए solvent front वाला distance तो same होगा, लेकिन the distance traveled by the dye would be different for each and every dye. For example, मैं इसको नाम देता हूँ dye A. Now the distance traveled by dye A was 2.1 centimeter. This is your numerator, जिसको हम कहते हैं distance traveled by the dye. तो ये आपके पास numerator आ जाएगा, which is 2.1. So 2.1 divided by 2.8 would give you 0.75. यही 0.75 is known as your RF value of that specific dye. This is how it is calculated. RF value क्या है? 2.1 divided by 2.8. 2.1 क्या था? Distance traveled by the dye. 2.8 क्या? Distance traveled by the solvent front, and hence the answer is 0.75. So if we talk about some of the applications of chromatography or advantages of chromatography, first of all, it is used to check the purity of a substance. Kyunki if I tell you one very important thing, how do you check the purity of any substance? Of water है, मैं आपको एक substance लाके दे दूं, I tell you that this water is pure. How are you going to check the purity? Purity क्या होती है? How pure is a substance without impurities? Water pure कब होगा? When there are no any impurities added into that water. So it is used to check the purity of a substance. Always a pure substance gives one spot on the separation. Moreover, it is used to separate and identify poisonous food additive or food colors which are not recommended by WHO. अगर आपने food additive या preservatives लोग कहते हैं कि यार this thing is not pure, ठीक है ना, इसके अंदर थोड़ा बहुत कोई ना कोई chemicals या preservatives added, तो chromatography is an application that can be used to check the purity of any kind of substance. Moreover, it is used to separate and identify even colorless compounds, for example, amino acids. Because we know biology students को पता है that protein is made up of colorless amino acids on separation. They are not visible. Then a locating agent ninhydrin solution is sprayed on chromatogram, due to which different amino acids becomes visible and different colors, which are then compared with the standard chart of amino acids, are hence verified. अब कभी कभार क्या होता है, a very common problem in chromatography is that some of the times the dyes are not visible. When the dyes are not visible, what you do is you spray a locating agent. आप क्या करेंगे? आप एक locating agent को यहाँ पे spray करेंगे. Now locating agent क्या है? वो locating agent की वजह से the dye would now become visible on the chromatogram, and the name of that locating agent basically is ninhydrin agent. Ninhydrin solution is the name of the locating agent which is sprayed on the chromatogram due to which different amino acids. अब amino acids की यहाँ पे example क्यों दी हुई है? क्योंकि amino acids जो है ना, वो जो colors produce करते हैं वो hardly नजर आते हैं. Because we will be when you go in A levels, you will be performing this practical inside your laboratory. A level chemistry में आप paper number 5 में ये perform करते हैं, paper number 3 and paper number 5 में of A2 chemistry में भी कि जब आप अमाइनो एसिड वाला एक्सपेरिमेंट करते हैं दे आर एक्चुअली कलरलेस डाइज, उनके कलर नहीं होते. दैट इज व्हाई यू हैव टू स्प्रे दिस मीन हाइड्रोजन सॉल्यूशन स्प्रे so they can be visible, and then you can calculate the RF value or determine the different number of dyes inside one solution. Moreover, this technique is also used by forensic scientists to investigate crimes from the incident place, and samples are collected which are then compared with the samples collected from suspects. If matching occurs, then they reach to a conclusion. Uses of chromatography question is very common. So basically in a crime scene, you can match your blood samples as well. Blood sample match करने के लिए भी forensic scientists use chromatography. Moreover, it is also used to identify poisonous insecticides which are not recommended by World Health Organization. Various plant pigments, for example, chlorophyll can also be separated by this technique. अब chlorophyll भी itself एक compound है. The students studying biology from me do know कि chlorophyll आपके पास chloroplast में एक compound होता है in plants which is used in photosynthesis. Now chlorophyll can also be separated by using the same technique of chromatography. And this is the end of your chapter separating techniques, and the last topic was chromatography, and with chapter number 1 of purification techniques, separating techniques and experimental chemistry भी इसको नाम दिया जाता है. This is all.